Today we are in the field in order to take soil samples for studying free-living soil nematodes. For sampling, we use steel cylinder with an exact inner diameter of 5 cm and an exact depth of 5 cm. The knowledge of the sample surface allows us to convert the number of nematodes we will count from the soil sample into the nematode density per square meter. The steel cylinder is hammered into the soil and then recovered with the help of a screwdriver. It is closed with lids from both ends and transported into the lab. We now arrived with our soil samples in the laboratory to start extracting the nematodes using the Baermann funnel method. First, we transfer the soil sample from the steel cylinder into a plastic bag and carefully mix the sample by hand through the plastic bag. The soil which we sampled today is relatively dry, so mixing is easy. We then determine the weight of the sample. Now we place the soil sample on a sieve with a milk filter. We use milk filters that are produced for milking equipment. These are standardized and have a high level of integrity, meaning that no fibers are released from the filter. Because the principle of the Baermann method aims to ensure that the nematodes migrate actively and as completely as possible from the soil, we only place part of the soil sample on the funnel, about 1 cm high. This subsample is also weighed and the mass is recorded as well. The ratio of the partial sample to the total sample is later also included in the conversion of nematode counts per subsample to densities per square meter. The soil is carefully and as evenly as possible distributed in the sieve. Any damage to the filter and any particles that get between the sieve and the milk filter will contaminate the sample. Therefore, it is important to work carefully. Sieve with milk filter and soil is then set into the Baermann funnel, which ends below in a PET hose, closed by a tube clamp. As soon as the soil sample has been inserted, work must be carried out with absolute care and without vibration so that no soil particles fall down in the funnel and contaminate the nematode suspension. Drinking water is added most cautiously into the gap between sieve and funnel, not through the soil sample, until the water touches the soil from below. This moment is easily recognized by a twitch and color change of the soil. Then immediately stop the water supply. After a few minutes, check again to see if the soil is moist and add more water only if necessary. Soaking the soil with too much water is not advisable. The soil only needs to be in contact with the water, then the paths between the pores are accessible to the nematodes. Now we have to wait for 3 days. Over the next 72 hours, the nematodes actively migrate downwards and then sink passively in the water column in the funnel, down to the hose clamp or into the glass tube. The first nematodes leave the sample because it is suddenly wet, later ones because it slowly becomes drier from above. After 72 hours, the extraction is ended by briefly and cautiously opening the tube clamp and taking the sunken nematodes with about 10 milliliters of water in a tube. If everything worked well, the yield is a clean water sample with thousands of worms wriggling in the clear water. For studying the nematodes alive, they can be placed directly with a drop of water on a slide under the microscope. For a more in-depth morphological examination, nematodes have to be killed. For this purpose, boiling drinking water is added to the nematode water in a ratio of 1:1. The abrupt rise in temperature from 20 to 60 degrees Celsius kills the nematodes in heat shock. Dying quickly, they relax and lie flat on their side in a position that is specific to the species and group. The shape can be outstretched or bent in different places of the body, from C-shape to helix. This shape is an important diagnostic feature for morphological identification under the microscope. For storage over longer periods of time, It is recommended to add 2 to 3 drops of formalin to the nematode water sample and store it in the fridge.